Okay, so we've learned a little bit about neurons and how they operate with a combination of electricity and chemistry. And now we can also begin to understand how the cortex is arranged into different areas that are devoted to different senses. So, for example, in the back of the brain here, in the occipital lobe here, the cortex in this region here is visual cortex because it is receiving inputs from the visual pathway. So here we can think of the retina down here as one of our sense organs, and the retina is going to transform light into electrical signals in the form of action potentials that will race down the axons. There's a synapse in a structure of the brain called the thalamus, and recall a synapse is where neurotransmitters are released and then channels open up in the target cell and ions go rushing in. And if those are positively charged ions, as they are here, there will be action potentials generated in this target neuron racing down the axons to the terminals here, and now we're up into the cortex. So this is the neural pathway that carries visual information up to the region of the brain, the region of the cortex that's going to further process that information. In this picture here, we see sort of a cross-section through somebody's head at the level of the eye, and so they're obviously looking forward here, and here we see the eyes, and out of each eye is the optic nerve. So 1 million or so axons carrying visual information from both eyes. This structure here is called the optic chiasm. Some of those axons are going to stay on the same side of the brain as that eye. Some of the axons are going to go to the other side of the brain. We'll come back to that later. Here we see a brain from the bottom, and some of the tissue's been dissected away. The eyes would be up here. Here's that optic chiasm. Here are the optic nerves continuing to go to the thalamus. So the thalamus we can think of right now as a kind of a relay station for the senses. So there's a synapse here, and then all of these white tracts here are the axons. So a bit of the brain has been dissected away, so now we can reveal the axons carrying visual information up to the back of the brain, the visual cortex. So this bit back here is the posterior region of the brain in the occipital lobe. So that's our visual pathway here. Now what we'll do here is learn a little bit about some famous experiments carried out some decades ago. And what the researchers were doing was measuring the electrical activity of the cells in this first cortical area for visual processing. They wanted to characterize what types of visual stimuli would activate those cells in the visual cortex. Okay, so here's the experimental setup to try and characterize the visual properties of these cortical cells. We have a cat here. It's been sedated, but it's awake, its eyes are open, and it's looking at a screen. And it's on the screen that the researchers are going to shine different kinds of visual stimuli to see if they can activate the particular cell they're recording from. So here they have a microelectrode inserted into the cortex, the visual cortex, and they happen to be right up next to a single cell, so they can record whenever that cell makes an action potential. They can record that electrical disturbance, and then they can kind of amplify it and give it a sound, so it'll be like a little click. So they can record the electrical activity of a single cell, and they just want to find what type of visual stimulus will activate that cell. Now, as it happens, it was in the early days of the research, it was really difficult to find some visual stimulus that would activate a particular cell that they were recording from. In fact, in this little video clip here, they would try things like just sort of a black dot like this, and they just couldn't seem to get the cell to fire. But then they noticed as they were putting the glass plate that had the dot here into the old projector to project it onto the screen, you'll notice they have to slide the plate in. So here comes and when they would slide the plate in or slide the plate out, the edge of the plate would pass through the visual field of the cat. And what they found was the edge was better able to stimulate the cell rather than the dot. And so just by accident, then, they happened to find a visual stimulus, an edge at a certain orientation that would activate the cell. And here's what it looks like with further investigation here. So here's a bar of light, and a particular cell that they might be recording from would respond maximally to a certain bar of light in that region, not somewhere else on the visual field, but in this region here, the cell would become maximally active. And if we had the audio turned up, you'd hear the clicking sound when the edge of light or the bar of light is at that location. So in the diagram here, we can see that's what this part of the diagram illustrates. So they turn the stimulus on at this time frame, so this is time, and then they're going to be measuring electrical activity, the action potentials in that cell they're recording from. Well, if they shine a horizontal bar of light on the screen, the cell was not active. It didn't generate a response. If they tilt the bar of light a little bit, still no response. But as they began to change the orientation of the visual stimulus, they got increasing activity until you get a maximum activity for a vertical edge or a vertical bar of light. And then again, if you continue to change the orientation, the cell gets quiet again. So the pattern of activity as they change the visual stimulus indicates that the cell that they're recording from, that particular cell, has a preferred stimulus of a vertical edge there. So a vertical bar of light will generate the maximum response, the highest frequency action potentials in that cell. So these cells came to be called sort of edge detectors or orientation cells, because when they would move their electrode, here's a cross-section of the visual cortex, when they would move their electrode at a sort of an oblique angle, they would encounter different cells along the path of their electrode and just record their properties. They would find, while the cell here was a vertical edge detector, you push the electrode a little further along and they'll find a cell that responds maximally to this orientation. Push it a little farther in, you'll find a cell that responds to this orientation. Push it a little farther in, you'll find a cell that responds to the horizontal. So there were populations of cells in the visual cortex here, receiving information ultimately from the retina. But these cells in the visual cortex seem to be tuned, seem to be maximally excited by edges of different orientations. So we can call these orientation cells or edge detectors. So, of course, it raises the question, why is it that cells in the visual cortex here, the first cortical stage of processing vision, why is it that those cells respond to edges? You know, how has that come to be? Why not spots of light? Why edges? And so when the neuroanatomists got into came on the scene here, they were able to do some tracing studies where they're going to sort of stain cells and see some of the patterns of connectivity between the retina and the thalamus and the cortex. They found something that looks like this. Okay, so in this diagram we see the wiring pattern from retina over here on the left to thalamus, and from thalamus up to the visual cortex. The visual cortex would include both the red and the green cells here. So recall that the retina, of course, is going to be where light is going to activate light-sensitive cells, and then the axons coming out the optic nerve will make a synapse in the thalamus. That's what you see here. So the thalamus would be represented by the blue cells here. So these black axons would be the optic nerves going to the thalamus. And then thalamus cells are going to send their axons up to visual cortex. And we saw that here. Here are the thalamus cells sending all these axons up to visual cortex. So the red cells and the green cells are both in the cortex, and the green cells are the ones that had this property where they were they maximally responded to edges of different orientations. So these green cells are the ones sort of pictured in this diagram up here. In this case, this is a vertical edge detector. Okay, so the way the system would work here is that we can think of the retina as a two-dimensional sheet. So it's flattened out here. It's really curved in your eye, but it's flattened out here to show that there is a kind of a two-dimensional layout of light-sensitive cells in your retina, right? And so they have spatial, relative spatial positions. There are some cells that are up here in the retina, some cells here, and over, left and right, and so on. Well, the way the optic nerve fibers run, there is a kind of a mapping, a one-to-one mapping between the two-dimensional layout of light-sensitive cells in the retina and the cells in the thalamus. So there is a preserved relationship, a spatial relationship between cells in the retina and the thalamus. Notice the cell up here in the retina is going to send its axon to a cell here in the thalamus, and a cell beneath it is going to send its axon to a cell also in the same relative position, you might say, in the thalamus. So there's sort of a one-to-one mapping, a preserved spatial layout between the retina and the thalamus. And now thalamus cells here are going to send their axons up to visual cortex neurons, the red ones here, which then also send their axons to the green cells. So to figure out how this system works, you can go two ways. You can either just sort of follow the visual signals from the retina to the thalamus and then up to the cortex, or you could work backwards. You'll notice each green cell has three terminals, 1, 2, 3, 1, 2, 3. And one of the assumptions we'll make here in this system is that this green cell will only respond when all three of its inputs are simultaneously active. That would be an example of spatial summation. The terminals are spatially distributed across the cell, but simultaneous activity would be enough to drive the target cell. Remember, neurotransmitters are released at each of these terminals, channels will open up, ions go in, and if enough positively charged ions go in, you reach that kind of threshold and you'll get the action potential generated in the target cell. So if we make this assumption that the green cells only respond when all of its inputs are simultaneously active, then we can figure out what type of visual stimulus on the retina would activate each cell. So again, now here there are two ways to do this. You could just say, okay, well, if we put a vertical edge or a bar of light on the retina, it means all three of these cells would be simultaneously activated, and then just follow the action potentials down their pathways and see where they end up. So if we take this one, the action potentials hit this target cell in the thalamus. This thalamus cell sends action potentials up to this one here, and that one sends action potentials down to this green cell. The middle one activates this thalamus cell, activates that cortical cell, activates both of the green cells, and this one down here activates that thalamus cell, activates this cortical cell, which activates this bottom green cell. In other words, a vertical bar of light simultaneously activating the three retinal cells in this vertical pattern here is going to activate these three thalamus cells, which end up converging on the bottom cell. So this cell will become active when all three of these retinal cells are simultaneously activated, and that is the case when there's a vertical edge or a vertical bar of light shining on the retina. This cell then becomes a vertical edge detector because it will respond to a vertical edge on the retina and not some other pattern of visual stimulus. On the other hand, let's use the reverse technique here. Let's take this cell then, find out what kind of visual stimulus it would respond to. Let's go backwards this time. So what you could do, since all three inputs have to be activated at the same time, you can just travel backward and see what kind of pattern of retinal activation would lead to simultaneous activity in the three terminals. So here we go. We'll just go back to this cortical cell, back to that particular thalamus cell. That thalamus cell is hooked up to this retinal cell. This terminal goes back to this cortical cell, back to that thalamus cell, back to the middle retina cell. This terminal goes to this cortical cell, to that thalamus cell, to this retinal cell. And so a diagonal edge would activate this cell. So the take-home message here was the way to explain why cells in the visual cortex had preferred stimuli that were edges, we can see that it's the wiring between the retina, thalamus, and the cortex that creates these cells such that they have preferred stimuli. So it's because the axons from three vertical cells on the retina end up converging on one cortical cell. That's what makes this cell's activity represent or stand for a vertical edge on the retina. Now, this wiring between retina, thalamus, and the cortex undergoes development, right? So this all has to be wired up during brain development. And so in the early days of visual research, after these orientation cells were discovered, scientists started to try to see if they could influence the wiring pattern of the early visual system during development. And they would use kittens in this research. So they would alter the visual experience of kittens. They could put goggles on them and so control what kind of visual stimulus the kittens would have for weeks at a time, and then they would record activity in the cortex to see if that altered visual experience influenced the kinds of cells they detected in the visual cortex. So in this case here, the kitten has horizontal goggles on, which would mean that the only visual stimulus that they see are horizontal lines, right? Horizontal edges. And if you do that, if that's the only visual experience that a kitten has for several weeks, and then later you record the preferred stimuli of cortical cells, what you find is far fewer vertical edge detecting cells. You find more cells that respond to horizontal edges and significantly less cells responding to vertical edges. Now, that was interesting because when the kittens kind of grew up with this altered experience, they did have trouble avoiding vertical edges like the legs of tables and chairs. And so they seemed to have a behavioral deficit. They didn't see vertical edges very well, and that behavioral deficit was related to the finding that they had fewer vertical edge detectors in their brains, right? So the biological explanation of their behavioral deficit is that their visual cortex did not wire up properly. They didn't have enough vertical edge detectors to give them the ability to see vertical edges in their environment.